crossover on that same chromosome. This is something known as interference. Okay, so if you think about it, we have our genetic distances here, and those genetic distances is nothing more than how frequently there should be a recombination event between those two genes. So 13% of the time there should be a crossover event between V and CT. 6% of the time there should be a crossover between CT and CV. So 0.8% of the time there should be two crossovers. We're just multiplying the frequency of the individual crossovers together. So if we look at how many flies we produced in total, from that table we had 1,448 flies. We should detect 12 double recombination events if those two crossover events are truly independent. If we went back to our data though, what we would see at that table is that rather than detecting 12 double recombinants, there were only 8. This is a very common phenomenon that you recover fewer double recombinants than you actually expect. And this is because of, like I mentioned, the phenomenon of interference. When there is one crossover on the chromosome, that actually makes it more difficult to have a second crossover event. And so we actually have a way of calculating it. The first thing we want to do is compare how many crossovers we observed relative to the number that we expected. And so in this case, we observed 8. We expected 12. And so what that means is we observed about 2/3 as many double crossovers as what we had expected. This is known as the coefficient of coincidence. Interference, then, the way to calculate it is just 1 minus that coefficient of coincidence. So if we take the number 1 and subtract the coefficient of coincidence from it, what that ends up being is 0.33, or in other words, we detected 33% fewer double recombinants than we expected to see because of this physical interaction of the chromosomes. Okay. This is an example of what a genetic map can look like when you're looking across number of chromosomes. So in this case, we're looking at a genetic map from Drosophila. They have 3 different autosomes and then 1 sex chromosome. And so basically what they've done is they've mapped the location of different mutations that are associated with different phenotypes. And so the name of the phenotype is on the right. And then on the left is the number referring to the genetic distance. So starting with 0, they just have the different genetic distances. So these are very common types of things you'll see with different things. Doing genetic mapping in humans is hard, right? Because we generate very few offspring. So what we have to do in humans a lot of times is to use pedigrees to help us. I'm not going to ask you guys to answer any questions like this because it's hard. This is kind of an example where we have the gene for blood type and a gene for a disease known as Nail-Patella syndrome. This is a dominant disorder. So what we see is this individual has the syndrome, and they have a capital N to show that they have the disease. Now, this individual is also heterozygous for blood type AO. Okay, so they have 1 copy of the A allele, and they have 1 copy of the O allele. Now, if those 2 genes assort independently, what we should see is that some of their offspring that inherit Nail-Patella syndrome would also inherit type A blood from that parent. But what we notice when we look at the pedigree is that every individual that inherits Nail-Patella syndrome from this individual has instead inherited the O allele from that parent. So it's the same kind of concept here where we see a correlation between the 2 phenotypes. And that correlation is due to those 2 genes being located on the same chromosome. That it's actually difficult. You have to have a crossover event to be able to pass the A allele of blood type with the N allele that causes the Nail-Patella syndrome. Okay. There are some other mapping techniques. By and large, we're going to come back to these towards the end of the semester. So I think in the interest of time, I'm going to kind of cut things off here, and we'll move into the group work stuff a little bit. Okay, so we're going to do a couple. There's only 3 problems. It's not too bad. 3 practice problems dealing with genetic mapping. How about, if you want to form up your groups, grab a paper, come down and grab it, and then once you're back to your seats, I want to give you some hints with question number 1 that will make your life a lot easier.